Hello students, welcome back. We're going to continue today with a new topic. We're going to be talking still about carbohydrates. So we're going to talk about carbohydrates and how they are carbohydrates when they're connected to something else, or glycoconjugates. And the first thing we're going to look at is carbohydrates in a bacterial membrane. No, not in a bacterial membrane, but in bacteria. Okay, I need to draw something for you to understand this. And this is how I'm going to do this here. Let's imagine that this ball here, this ball is our bacterium. And some bacteria is our bacterial membrane. Some bacteria have a layer or a sheet of carbohydrates and proteins, or carbohydrates and peptides. And so let's say that this sheet, this represents that sheet. And so some bacteria have one sheet and some have multiple sheets. And so the first thing we need to do, though, is move our bacteria out of the way. First thing we need to do is identify what's found in this sheet. We need to draw this sheet first, and then we'll use the sheet. Once we know what the sheet looks like, we'll be able to distinguish the two types of bacteria. And so let's draw the sheet first. And the carbohydrates that we'll use are carbohydrates that you're very familiar with. Okay, and let's draw this first sheet here. Now, the first sheet, let's see here. I'm going to actually use, orange as my color. The first sheet is made up of, okay, we're going to put here NAM, N-acetyl muramic acid, and we know what muramic acid looks like. We've already seen that one. And N-acetyl muramic acid is going to be connected to another sugar, N-acetyl glucosamine. And so that's kind of going to be our disaccharide unit that we're going to use to make this sheet that goes around the bacterial membrane. So we're going to have N-acetyl muramic acid, NAM, N-acetyl glucosamine, NAG. Then we're going to repeat that. N-acetyl muramic acid, and acetyl glucosamine. Okay. And that's going to just continue. So that's going to continue this way, and this one will continue this way. Okay. And so that's just one line of our sheet. So we're making the flat sheet right now. Okay, and then we'll have another one of these. And now, since you know what these are, the rectangles will represent NAM, and the hexagons will represent NAG. Okay, and we have another one, and then we're going to have another one. We're just drawing our sheet right now. And this is going to continue on and on forever. And so, these are our sugars. Okay. And so we can call this sugar. This is our glycan backbone. And so this is our glycan backbone. So we have our glycan backbone. So we have these long strings to make a sheet. Okay? But they're not connected. So strings need to be connected to. They need to be woven together. The strings need to be. Now, attached to each of our NAMs, our N-acetyl muramic acid, is going to be a peptide. And we'll make the peptide. a tetrapeptide. So it's going to go here. 1, 2, 3, 4. 1, 2, 3, 4. 1, 2, 3, 4. And so we're going to have the same thing attached to all of our NAMs. So we have NAM nag. Oh, let me erase that one. It's going over a little bit too far. 1, 2, 3, 4. 1, 2, 3, 4. And 1, 2, 3, 4. Okay. And so we have now a tetrapeptide. And so I'll just make this orange again. And so this is our tetrapeptide because it's 4 amino acids. A tetrapeptide. And again, what we're trying to do is make the sheet that goes around the bacterial membrane. And I'll explain why do we need a sheet around a bacterial membrane. I'll explain that in just a second. But now what we have to do, we need to weave these together. And to do that, there's a cross link between each of our strings, each of our glycan backbones. And so what happens here, the third peptide, let me make sure I get these numbers right, because this is kind of important. Yes, the third peptide here, the third amino acid is going to join with the fourth here, and the third here is going to join with the fourth here, and you're going to tie all of these. You're going to tie all of these strings together, and you're going to weave a sheet. And this is our sheet. And this is our cross link here. Okay, and so in our sheet we have 3 components, I think. We have our NAM-NAG glycan backbone. We have that. We have our tetrapeptide made of 4 amino acids, and we have our cross-link. And so this is our sheet. And before we continue, we're going to go ahead and give a name to this sheet. This sheet is called a let's get a good color because this is important. This sheet is called the peptidoglycan. The sheet is called a peptidoglycan layer. A peptidoglycan layer. Okay, we're going to use that sheet. That's what we've just created, a sheet. And this sheet, and this is the membrane of our bacterium, this sheet here is going to cover that bacterial membrane. Some bacteria only have one sheet. Some bacteria have many sheets. Okay? And that's what we're going to draw next. But we're going to stop on this slide. And so we have our peptidoglycan layer. And that peptidoglycan layer is going to be very important in distinguishing the 2 major classes of bacteria. Okay, now that we have our sheet made, let's look at the 2 different types of bacteria. Okay, and I'm going to move this over here. And what I'm going to do, I'm going to make the membrane here, the bacterial membrane. I'm going to make it blue. So this is going to be my bacterial membrane here, and I'm going to make my sheet. I'm going to make my sheet orange. And so there are some bacteria that have a sheet here, and they have outside of that sheet, they have another sheet, and outside of that sheet they have another sheet, and outside of that sheet they have another sheet. And so some bacteria have multiple sheets. Some have multiple sheets. Okay. And let's look at and there are other bacteria divided into two groups. One with many sheets, and there's another type of bacterium here. And this bacterium only has one sheet. So here we have, and this is important, this is very important in terms of classifying the two groups of bacteria. These bacteria have multiple sheets. And on the right, these bacteria have no multiple sheets. There's only a single sheet, so no multiple sheets. Okay, so we have multiple sheets on the left, no multiple sheets on the right. In addition, let's look at the bacterium on the left. This bacteria has an additional membrane outside of its peptide glycan, or its peptidoglycan sheet. And so on the left we have bacteria that have multiple peptidoglycan sheets, and on the right we have bacteria that have only one peptidoglycan sheet. Because the bacteria on the left, because they have multiple sheets, you need something to hold them together, hold those various sheets together. And you're going to have this structure here. I don't know what would be a good color. I guess green would be a good color to use here. There's going to be, it's going to be carbohydrate in nature. There's going to be an acid here that holds these multiple sheets together. And we'll learn about its name later. In addition, and so this structure that holds those multiple sheets together is not found in the no multiple sheets bacteria, because it would be pointless because there's only one sheet. However, I'm going to make this second membrane a little thicker because we need to see something. Let's make this membrane thicker over here because we need to put some things in this membrane. Okay. In this membrane, the outer membrane of the right bacteria, there's going to be there's going to be a disaccharide. Okay, there's going to be a lipid. I'm going to draw the lip and try to think of a good color here. Okay. Well, I'm going to put the disaccharide here. There's going to be a disaccharide in this membrane, and then a lip anchored with lipids. Disaccharide anchored with lipids. Disaccharide anchored with lipids. And that's going to be in this the membrane of these bacteria. And connected to those disaccharides, it's going to be a very long polysaccharide. A very long polysaccharide. Okay? And all of this structure here has a name. We'll learn about what this is. So this is going to be a polysaccharide. And we're going to have here a lipid. Okay. And those are all of the components of the two types of bacteria. Now what we're going to do, I'm going to give you the names of the two types of bacteria, and then we're going to label all of these components. Okay. So we know, and I'll do all of my labeling in black. That would be a good color. So we know that this is our peptidoglycan. We've seen that. That's our peptidoglycan. Now, on the left here, these bacteria have multiple sheets or multiple peptidoglycan layers. And so these bacteria are called gram-positive because there's a stain, and that stain stains these multiple layers. So these bacteria are called gram-positive. And they're called gram-positive because they have multiple layers. And on the right here, these bacteria, they only have a single layer. So these bacteria are called gram-negative because they don't stain. They don't stain the same way. So they're called gram-negative bacteria. These green structures here that hold the multiple layers together in gram-positive bacteria, these green structures, this is an acid called teichoic acid, T-E-I-C-H-O-I-C, teichoic acid, and it holds these sheets. It holds these sheets together. And so teichoic acid is going to be found in gram-positive bacteria, but of course it's not going to be found in gram-negative bacteria. And again, teichoic acid is a polymer of sugar alcohols, and so alcohol sugars. Okay, and so again, carbohydrates are found there as well. In your gram-negative bacteria, you have these long structures here, these very long structures that protrude, that extend off of the outer membrane. This is called the lipopolysaccharide. Lipopolysaccharide. Now, lipopolysaccharide can induce a very strong immune response. When gram-negative bacteria, when they get into your bloodstream and they're destroyed, these lipopolysaccharides are LPS. This LPS is released, and that causes a very strong immune response, and that can lead to septic shock. Septic shock. And this is how gram-negative bacteria can kill you, due to septic shock. And lipopolysaccharide has another name. It's called endotoxin. And the rationale here is that this is the toxic part of the bacteria, but it's integrated within the bacterium, and it's not released, and that's called endotoxin. Gram-positive bacteria, however, they don't have any endotoxin, but they can release exotoxins. They release exotoxins. And this is how they exert their effect. Exotoxins. And some exotoxins include things like, I think, ricin A is an exotoxin. Tetanus toxin. Tetanus toxin is an exotoxin. These are proteins. What else? Oh, cholera toxin is an exotoxin. Cholera toxin, pertussis toxin. And these are all exotoxins released by gram-positive bacteria. So, just to review, on the right, on the left rather, we have our gram-positive bacteria, and the gram-positive bacteria have multiple peptidoglycan layers, and they have teichoic acid to hold the layers together. On the right, we have gram-negative bacteria, and the gram-negative bacteria only have one peptidoglycan layer, and so there's no need for teichoic acid. Gram-negative bacteria have two membrane layers, an inner membrane layer and an outer membrane layer. In that outer membrane layer, we have lipopolysaccharide. And that lipopolysaccharide is also known as LPS, or endotoxin, and this can cause septic shock. And a lot of this kills a lot of people in ICU, especially if they have septic shock. And so these are our two major classes of bacteria, and that's all we're going to say about those. I just wanted to show you how carbohydrates are important in classifying bacteria and important in the cell wall of these bacteria. Oh, and just so you'll know, this, where you have the peptidoglycan layer, that's the bacterial cell wall. So gram-positive bacteria have a very thick cell wall. Gram-negative bacteria have a very thin cell wall. And so these peptidoglycan layers make up the cell wall. And that's all we're going to say about these here. And on the next slide, we'll just make a table looking at the difference between the cell walls of gram-negative bacteria and gram-positive bacteria. Hope this makes sense to you. On this clip, we're going to just look at, we're going to make a chart and to compare our gram-positive bacteria and our gram-negative bacteria. But remember, it's carbohydrates that are mainly responsible for these distinctions. And let's make our chart here. And so here we're going to put gram-positive here. I'm going to just put gram plus and gram minus. I don't need that much space. I'm going to just do this over again. I'm going to put gram plus and gram minus, because I don't need much space for these, but we'll need a lot of space for the one on the left here. And the first one, okay, the first thing we're going to look at, so let me make it a little straighter, multiple peptidoglycan layers. We'll look at that first. Multiple peptidoglycan layers. Okay, we're going to look at that first. Secondly, we're going to look to see the number of membranes. Number of membranes. How many membranes are there? Thirdly, we'll look at the presence of teichoic acid. Is teichoic acid present? Or teichoic acid? Next we'll look at, we will see, is endotoxin present? Endotoxin. And that'll be LPS. LPS. Is it present? And lastly, we're going to see, does it secrete exotoxins? Does the bacterium, or do the bacteria secrete exotoxin? And we've seen examples of the exotoxins, things like cholera toxin, ricin A, tetanus toxin, diphtheria toxin. All of these are exotoxins. Okay, and here these are going to be our, these will be our cell wall features here. Okay, now let's fill in our chart. Multiple peptidoglycan layers. And of course, for gram-positive bacteria, that's going to be yes. For gram-negative bacteria, no. And this is what makes this is the main difference between gram-positive and gram-negative. If there are multiple peptidoglycan layers, then it's going to be gram-negative. Okay. Next, let's look at the number of membranes. In gram-positive bacteria, there's only one. Gram-positive bacteria, there's only one membrane, whereas in gram-negative bacteria, there are two membranes. Thirdly, teichoic acid. Is teichoic acid present? Remember, the whole purpose of teichoic acid is to hold those multiple layers together. So gram-positive bacteria would need teichoic acid since there are multiple peptidoglycan layers. However, gram-negative bacteria would not need it because there's only one layer. I know all of you all have taken microbiology, so forgive me for going over this, but it is very important to understanding the role that carbohydrates play in these in the classification of these bacteria. Endotoxin. Is endotoxin present in gram-positive bacteria? It is not present. In gram-negative bacteria, it is present. And this is what causes things like this is what causes septic shock, because when the bacteria are destroyed, it releases these endotoxins, and that causes a hypersensitive immune response, and that's what causes toxic shock almost, or causes septic shock. That's the better term, septic shock. And lastly, is exotoxin present? And in gram-positive bacteria, you do have exotoxin. Now, I'm making this one red because you would think, well, since gram-negative bacteria has the endotoxin and gram-positive bacteria does not, then gram-positive bacteria should have exotoxin, and gram-negative bacteria should not. However, both produce exotoxins. So just remember that I made that one red. And so I hope this little graph here will show you the key differences between gram-positive bacteria and gram-negative bacteria. Again, I know you've taken microbiology and you've gone over this, but I just need to go over it with you to make sure you understand this in the context of biochemistry and carbohydrates. And that concludes this